Welcome to this interactive whiteboard explainer. I'm super thrilled you're here. Today's session is meticulously designed to secure your vital marks in O-level biology genetics. We're going to focus specifically on the real intricacies of human blood groups and plant crosses. We are literally going to decode codominance and multiple alleles, visually mapping out the exact mechanics on screen, so you can master these concepts completely. Let's get to it. Let's hook right into a real-world macro puzzle, okay? Have you ever wondered why some blood is AB? I mean, really think about it. Why do some people possess a blood type that isn't simply A nor simply B, but instead it's a full-blown expression of both? This whole puzzling phenomenon is honestly the perfect gateway into understanding how certain genes interact when, well, neither one is willing to step back. So here is our exact syllabus for today's whiteboard session to map out these complex genetic rules. We're going to hit the mystery of AB blood, then decoding codominance mechanics. After that, mapping multiple alleles, followed by step-by-step genetic crosses. We'll wrap it all up by spotting the common mistake, and finally, your exam tips and recap. All right, Section 1, the mystery of AB blood and how we are moving way beyond Mendel's P's. Moving right along to Section 2, decoding codominance mechanics, where both traits are fully expressed. Okay, so what actually is codominance? Well, it's a genetic scenario where both alleles are fully expressed in a heterozygous organism. Let's look at the screen. Take this oval leaf here. If we magnify its microgenetic, like in that little pop-out bubble, you can see exactly what's happening inside the cells. We've got the capital R for the round leaf allele right next to the capital L for the long leaf allele. They're sitting right there, side by side. Look at the call-out tags. Neither allele is masked. Both the R and the L are fully expressing their instructions simultaneously to create that unique oval shape. That, my friends, is codominance in action. Next up, Section 3, mapping multiple alleles across the population pool. Now, this is absolutely crucial to grasp. Individuals carry exactly 2 alleles, but populations, they can hold 3 or more. If you look at the individual level, you, me, we only have 2 literal slots for our genetic code. But if you zoom out and look at the larger population pool, that single gene can contain more than 2 distinct forms or alleles circulating out there. It's kind of like a giant genetic library. The classic textbook example of this is the ABO blood group system. As you can see mapped out here, we have 3 distinct alleles in the pool: IA, IB, and little io. In this specific system, IA and IB act as these codominant heavyweights. Notice the vectors showing how they both completely dominate over that recessive IO allele. But when IA and IB are paired together, just like we saw with the leaf, they stand side by side, perfectly equal in their expression. All right, let's get practical. Section 4, step-by-step genetic crosses, doing the math. Let's actually solve a problem involving those codominant plant alleles we looked at a minute ago. We'll use a standard Punnett square. We're crossing 2 heterozygous oval-leaved plants, so that's RL crossed with RL. Follow along with me on the board as we track the flow of these exact variables. Looking at the top row in the left column, we've clearly labeled our parent alleles, R and L. By mapping this cross, dropping the columns straight down, and dragging the rows right across, we can explicitly track how the parental traits transfer into the offspring cells. Look inside the grid. We end up with 1 RR for round, 2 RLs for oval, and 1 LL for long. And boom, that brings us right to this resulting phenotypic ratio, 1:2 to 1. 1 round, 2 oval, 1 long. This is the direct mathematical footprint of a codominant cross between 2 heterozygotes. Whenever you do the math on a codominant cross like those leaves, and you land in a 1:2 to 1 ratio, you know for sure your calculation is right on the money. Now let's step up the complexity just a bit to a multiple alleles scenario. We'll follow this flowchart step by step. Step 1, we identify the parent alleles. We have a person with blood type A, specifically genotype IAIO, crossing with someone with blood type B, genotype IBIO. Moving to step 2, we map the cross using our trusty Punnett square. And step 3, we reveal the offspring diversity. This is wild. These parents can astonishingly produce children with any of the 4 possible blood types, A, B, AB, or O. That massive diversity from just 2 parents, that's the direct result of multiple alleles at play. Moving on to Section 5, spotting the common mistake, codominance versus incomplete dominance. Let's compare these two directly on screen, because this is where a lot of people lose points. In codominance, both alleles are fully expressed side by side.
Think back to those R and L leaf alleles, both distinct, both present. But in contrast, with incomplete dominance, alleles blend together to form a mixed phenotype. Never ever confuse the two. Codominance is the distinct, simultaneous expression of both heavyweight traits. Incomplete dominance is a mere blending. Actually, it's more like mixing red and white paint to get pink. Keeping this conceptual boundary clear is absolutely critical for your exams.
And finally, Section 6, Exam Tips and Recap. Let's secure your vital marks. Let's run through our quick-fire explainer recap. First, codominance. That's the full expression of both alleles and heterozygotes. Second, multiple alleles, meaning more than two forms of a gene exist within a population, even though an individual only ever holds two. And third, the ABO blood groups, literally the ultimate example of both multiple alleles and codominance working together. Keep these quick-fire rules anchored in your mind. I really want you to mentally visualize that magnified oval leaf and the dominant blood type vectors for instant memory retrieval when you're sitting in your test. And here is a non-negotiable exam tip for you. Always practice drawing Punnett squares to visualize your outcomes. When you get a genetics question, read your exam prompts carefully to verify the exact inheritance type. Is it codominant? Is it multiple alleles? Once you figure that out, physically draw the grid. Label your columns, label your rows, and prove your math step by step. Seriously, never try to hold an entire genetic cross in your head. It's a trap.
So I'll leave you with this final kind of provocative thought. If you know a couple's blood types, can you perfectly predict the genetics of their child? Well, we just saw how a type A and a type B parent can actually have a type O child. The variables are just fascinating. So I highly encourage you to grab a piece of paper right now, set up a Punnett square, and try solving the mystery of your own family's genetics using the exact mechanics we mapped out today. Keep practicing those visual grids, and I will see you in the next explainer.